So it is our pleasure to introduce Professor Ulf Kalhert from Otto von Guericke University, Magdeburg in Germany. He's actual head of the Department of Molecular and Experimental Surgery, but he's also an international expert in translational cancer research with particular expertise in biomarker discovery for prognostic and therapeutic applications. He's working in high-volume academic surgery department with a strong translational research. And today he will share with us his perspective on liquid biopsy diagnostics for perioperative care in pancreatic cancer, insights from East German Academic Surgery Department. Thank you very much, Ulf, for joining us.
Yeah, dear hosts, Bruno and Ingrid, and dear Transplant members and affiliates, thanks for the invitation. Maybe a brief explanation why I put East German here. You know, we have a one country. This is due to the fact that the aging population between East and West is not necessarily different, but the dynamics is pretty different, and the composition is also very different. That is due to certain facts of immigration dynamics, more to the west, less to the east, but also, East Germany had a different system for 50 years, and this influences also the health sector and the democratic composition of the people. So we have usually to deal with an average older population in rural areas in Germany than in West. And as we know, pancreatic cancer is also aging disease. Mostly, this has some relevance, I think. That's why maybe this is the short outline why East Germany. Yeah, and brief introduction. The clinic. I have the full professorship for research, and Professor Kroner is the clinical head of our department, the director, and this is a high-volume surgery center for our state of Saxony-Anhalt. I come to this later. And we actually clinically acting on three. Can you see the mouse? No, probably not. I'll try to figure out the pointer here. We're acting on three institutions. So the clinic is spread over three physical sites, and you see the structure. We have over 125 beds affiliated to our clinic, and a certain highlight is certainly that we have three dedicated operation theaters for our clinic, and two surgery robots, including the latest version of the intuitive surgical devices, Da Vinci. We have an oncology certified center. That means certain quality of the clinical care, certain quality of the education of the doctors, and also current activities in clinical studies. Here we come to our research focus also is involved. And yeah, currently we have over 40 doctors with 1/3 of higher senior attending doctors, and we have inpatient cases of 4,000. So the spectrum, I can go broadly. It's on the surgery. It's from the upper GI to the lower rectum, essentially. Every acute case and also scheduled case is taken care of by dedicated sections. It's almost like an American system with sub-departments on the HPB, on the colon, and on the upper GI, the esophagus. Yeah, and this is the setting where we are coming from. Another resource I would like to share is the long-running registry study of patients operated in our center, but also in cooperating centers. At the highest time, we had over 400 centers affiliated to this study. And essentially, with a Q&A reporting system from the surgeons, we register pre, intra, post-op clinical events. And this can be done in a very thorough way, but also sometimes very superficial. So the cases are relatively high. Currently, we are counting of almost 100,000 colon cancer patients that are, registered like this, and also a good amount. It's a rare disease, the pancreatic cancer patients. Where we record these essentially quality parameters in the operative care. And a goal that I have is to fuse all of these cases with molecular data. And we are started now with putting histology data to the clinical cases for the pancreatic cancer with a funded project. And this has nothing to do with liquid biopsy. So then hopefully it's working. Thanks for the interruption and noticing. Yeah. I see the role of my department basically in bridging these, high volume clinical care to the coordinated research deliverables, and the aspect we are going here is essentially reverse translational research. And another fact which I touched very, shortly in this presentation is I do see at least 30% of my activities, of our activities, in robust infrastructure development for surgical contexts. And here we mention not only the biobanking, but essentially these data set that we retrieve from the surgical care givers, but also from us by sequencing the material associated to the individual specimens. And translating this essentially in innovations. We focus here on our visibility of the clinic with third-party funding and network activities. North Saxony-Anhalt, the population here we see is the area that we take care of. I'm not going into detail. It's a very old population, amongst the oldest in entire Germany, with 2 other states. And on the lower right, you see Leipzig and Halle taking care of the south part of the state. The research I'm sharing today is always in close and actually also in the involvement of our national patient advocacy group, patient representative group. It's the ADP, so-called Arbeitskreis der Pankreatomierten. This is for people who received operations on the pancreas, not only cancer. And this is an important fact from us. We have regularly meetings with them, including the national convent from them coming up in May. And with this, we not only work on the same news outlets and networks. No, we really would like to also have the opinions about our deliverables in a close connection, not only in the course of a grant application. We really develop these symbiotic messages in almost all projects with the pancreatic cancer as of now. What is the clinical problem for pancreatic cancer? We all know it. I don't think in this audience I have to spend so much time on this. It's not only very aggressive and very hard to treat disease, which may be the case because it's also late detected, but this is the most important point, is essentially difficult to diagnose in an early status. Patients occur in an advanced stage with highly symptoms, and this is essentially the working paradigm of liquid biopsy. We can catch the tumor earlier, or we can catch tumor remission, failure of tumor control after the surgery better than with the standard clinical diagnostics at this moment. And for us as a surgery department, I focus here this schema, which we usually use in presentation, is late detection. Most of the pancreatic cancers at the detection stage are not directly operatable, which is still the treatment option of choice in terms of positive prognostic value. You have to admit that many patients receive chemotherapy in a neoadjuvant setting, and we know the effects are not only on the cancer. The quality of life is severely diminished, and only then, if cases respond, they can, in a good fraction, luckily, but not in a very high fraction, benefit from surgery. And yeah, if you can detect tumor earlier, we also believe that we can catch the tumor in a stage that can directly benefit from surgical interventions. This is our working plan. Yeah, why? What's the clinical standard at the moment? We know that liquid biopsy is based on CA19-9, the glycan positivity. Different nations have different threshold levels on how much need to be detected, but this is essentially a yes or no situation. And of course, the clinical symptoms and the medical imaging. Yeah, without going into details, but I think a benefit of a liquid biopsy, other than the glycan CA19-9, which is also associated to some inflammatory diseases and not necessarily tumor disease, have benefits in improving the entire sensitivity and specificity than what is currently used. We know this is a high bar, and the large-scale multi-center phase 3 clinical trials have to prove new liquid biopsy markers to really be specific and sensitive, especially for such a rare disease of pancreatic cancer. You need to have almost 99% of specificity to make it approved by the healthcare providers. But I still think the current clinical used diagnostics lack on specificity. And this is another working hypothesis why we think on liquid biopsy. Yeah, just I'm not sure how many people really work on liquid biopsy. I think this is a major breakthrough that is focusing on the cell-free DNA, essentially tumor DNA shed from dividing tumor cells, or maybe it's also another reason why they shed this, but at least we believe that this is highly tumor associated because we look for mutations that tumor is defined by our current NGS based diagnostics. And this is a paper in the context of colon cancer, and basically what they analyzed is for low-grade colon cancer, where it's still the question whether the patients really benefit from adjuvant, so post-operative chemotherapy with all its side effects. So the investigators compared the decision on post-op chemotherapy based on a certain detection threshold of circulating tumor DNA that defines the colon cancers. In this case, I think it was also RAS mutations and BRAF mutations, versus the standard of care. What is currently done is looking at the tumor area in the medical imaging with the CT. And essentially the bottom line from this paper was that management of chemotherapy by circulating tumor DNA reduced the fraction of patients that need chemotherapy. You see it here, 15 to 28, but the overall survival of these patients in this group was not diminished. So in other words, this is a good application in a large trial where circulating tumor DNA in the GI cancer field can help the patient. In this case, not with the survival, but with the life quality. And essentially also the pharma industry with, maybe not spreading the drugs too much where it's not needed. Yeah, and this is a 3 years old paper. Many things are active. I take a few papers that I think are important. It's essentially a follow-up of this study published in Nature Medicine last year on the long-term survival. It looks still, supporting the notions that I just mentioned to you. And now the investigators go into these patient management in later stage colon cancers as well. And yeah, one paper I found recently is on the testing the efficacy of the new KRAS inhibitors in the field of pancreatic cancer. There's not much so dedicated circulating tumor DNA clinical trials ongoing in the pancreatic cancer field to my research, probably due to the fact that it is relatively established now in the clinical QL field, and mostly the pharma industry is already using it to tailor down their their drug efficacy. So it's essentially only used already as a continuum diagnostics. Yeah. However, you can also say that the therapy developments that are very emerging and very focused and very fascinating for pancreatic cancer and other very late stage cancers using these liquid biopsy fields. So the summary from this slide is essentially that this is now a more or less standard method that is used in clinical trial development for drugs. Here is on the left side is the ASCO GI abstract from the KRAS inhibitor study that now an update has been provided in ESMO also on colon cancer. So people monitor treatment response with the circulating tumor DNA quantity. And also in the pioneer paper on of the neoantigen vaccines, the BioNTech trial, at least the investigators noted that they used the circulating tumor DNA to assess the response. However, I couldn't find it in the original data nor in the supplements. So maybe it's coming up in the follow-up publications. So this is essentially the general interest. And yeah, our lab, we focus on microRNAs rather than circulating tumor DNAs, mostly due to the fact that we cannot compete with large consortia pharma industry studied, and also from a basic fundamental science standpoint of view. There are from basic science credible evidence available that epigenetics acts earlier than genetics. So microRNAs are epigenetic regulators. This is an aspect one can use as an argumentation. We also noticed from other studies that microRNAs can be specifically used as a diagnostics in a multiplex fashions versus the circulating tumor DNA mostly focuses on one or maximum two targeted sites that define the tumor mutation. Yeah, we also think that microRNAs particularly act very dynamically on changes on the microenvironment versus the tumor DNA comes from a mutated cell, so the tumor is relatively established. So I think it's also a same thing like epigenetics versus genetics is a relatively later level of tumor detection. Yeah, the argumentation the microRNA field faces is stability. RNA, we know in circulation is almost unstable on long term. So how can you really use this as a diagnostics? Here I will showcase some study where we think on extracellular vesicle encapsulated microRNAs can save as a protection and thereby overcome this stability issue versus DNA is much more stable. And of course, specificity, I mentioned it, only large scale multicenter randomized phase 3 trials need to prove this. Yeah, in the first study that I want to showcase here, we have a strict timeline. I will just give you the summary. Here is where we used different machine learning algorithms on sequencing material from our patients and validated this with publicly available data sets to identify a microRNA panel of these 3 microRNAs depicted on the right side versus the 205, we believe, is a very, very interesting marker and has come up actually in another cancer field, and the industry is following up on this, lung cancer, as an early detection for treatment failure. And we have identified those microRNAs. Briefly to the study is a relatively robust approach. We just took the tissue from our patients and with the help of the pathologists separated into tumor versus non-tumor from the same patient, non-tumor based on the Sentinel method. Sentinel method is a very clean and very precise method how the pathologist looks at a certain tissue. It's not the current standard. So we had to have a specific ethic vote because the patients would probably receive a better diagnosis than those that are not enrolled. Essentially, we you looked very carefully, the pathologist, into the tissue and couldn't find tumor cells. So the resection margin without tumor cells is defined as non-tumor tissue. And we took patients' blood. In this case, we've utilized the serum pre-operation, so where the tumor is still in full mode. And we also took particularly care that we collected the samples before the onset of the anesthesia, as we know that this is a very traumatic event for the patient. So that also changes the microRNA abundancy in the blood, I think. And we essentially sequenced this material, blood and blood serum, and did almost a subtraction-based method, and identified essentially differentially expressed microRNAs that are upregulated in tumor versus non-tumor, and then looked of those tumors, differentially expressed genes that are upregulated, if we can find those in the patient's serum, and then narrowed down essentially target or candidate targets, and then by integration of publicly available data sets, we narrowed down through these 3 microRNAs, where we particularly found that the microRNA 205, when you look into the tumor tissue itself and clinical data, is itself as a prognostic marker in the tumor, but it has not been described as a liquid biopsy marker. And currently we're following up on this. We received the funding. We're doing a multi-center prospective validation trial for this triplet. And I want to focus that we really think is a nice marker to monitor tumor recurrence in the patients. Whether that can be used as a discriminator of pancreatitis versus tumor, whether we can use this as an early detection, who knows? We're focusing on the post-operative therapy monitoring. Another concept that was introduced in a first publication from another study, but I want to showcase this also in the context of microRNAs and liquid biopsy tool, is the low abundancy microRNAs. Mostly also the study that I showed you just now uses on what is maximum expressed, what is maximum differences, differentially expressed between 2 groups to compare. If you look in the proteome field, especially from model organisms, fruit fly and zebrafish, the concept of low abundancy proteins as a key regulator of physiology describes essentially that certain high potent proteins involved in cell cyclus regulation or stress responses have a very tight control in their regulation, particularly during aging. And once a certain aging is reached, or age level is reached, these proteins are essentially shut down forever, and only in an unknown or unwanted event like a disease progression, these low abundancy proteins are essentially dysregulated and then activated, but not to a humongous way, just a little bit. So essentially these low level differences of very important proteins are called low abundancy proteins, and we focused on low abundancy microRNAs by a definition only on the expression level that we identified in our data sets, the lowest 30% of all recorded signals. And by essentially doing such a mathematical attempt, we proposed a new microRNA triplet that has an utility to detect pancreatic cancer from non-cancer. Briefly guiding you to this multi-center trial that was also recognized in ESMO last year to be presented. 2 German center studies with 109 patients provided us, including 1 center is from us, essentially our patients, RNA-seq data, and we integrated these differences between pancreatitis, chronic pancreatitis, and pancreatic cancer, low abundancy microRNAs in publicly available data sets, mostly from Japan and Korea. So it's essentially a bioinformatical project with over 4,000 patients, and we narrowed down target candidates, which I don't reveal because we currently submitted invention report, and maybe there can be a patented scenario out of this. It is ongoing. It's also a funded project here in my lab. where we do now the validation of these microRNAs in a prospective fashion in 2 centers, one in China and in our centers. And we have already enrolled a good amount of patients now, where we test these microRNAs, and the results are not available there. So I think this is a new concept, low abundancy microRNAs, as mostly unrecognized part of the microRNA transcriptome to maybe help tumor detection or disease detection in general. And we've exemplarily showed you on the pancreatic cancer. Yeah, this is an outreach. This is without the prospective validation. This is only the retrospective data that our panel is, in terms of AUC performance, outperforming the clinical marker that is currently used. But again, these are retrospective patients from selected study centers. So we want to continue our efforts and show the results in a prospective validated fashion. Yeah, in a small project, I also see this is not yet published. This is where I participated as a cooperation partner, essentially with the Transpang. It was initiated, I must say, thanks for this, with Professor Pina Onungwa in London. We sequenced an age-matched small scale clinical study, but small scale. Why? Because we have coincidental findings of pre-malignant conditions in our clinic, so IPMN in particular, and we sequenced essentially the blood from those patients and did a differential analysis and propose a few candidates which may help to catch pancreatic cancer in development, because we did a bioinformatical approach to identify trends of microRNAs from the few IPMN cases over chronic pancreatitis to the pancreatic cancer. We will see how this holds for publications, but this was an attempt because we had coincidental findings in our clinic of IPMN, a case that is not very often detected, in particular not that they are enrolled in a study where they approve the biosampling of their blood. So this I just wanted to showcase you. And then in another study we published in eLife, I think last year, is where we used, so to say, a very known and very powerful type of diagnosis in cancer and pancreatic cancer, the radiology. Although we know it is not easy to discriminate a tumor versus a pancreatitis, but we essentially stratified patients based retrospectively on the radiology, and then also, because the patients were operated by pathology confirmed, stratification in chronic pancreatitis and pancreatic cancer. So these images on the left side may be very difficult for us to discriminate, but after the operation and the histology assessment, we know what was a tumor and what was not a tumor. And from those patients, we also sequenced the blood, but not the whole blood. We separated the extracellular vesicles fraction from this simply by ultracentrifugation and size exclusion. We didn't focus on any marker selection. I mean, this is also a very, very difficult and very dynamic field. We did a relatively robust attempt as a surgeon department, just collecting and spinning down, and of course verifying that the markers expressed on this surface of extracellular vesicles are there, but never used any epitope-based selection. And long story short, it is published. microRNA panel was described that helps maybe to identify our tumor versus a non-tumor in the context of pancreatitis separation. But the build-up of this story was that we used the radiological features from these patients. So in a outlook and in a application-oriented fashion, we thought that rather than only focusing on blood-based diagnostics, we also selected certain features from the radiological images and maybe help by computer vision, essentially, the radiologist to better discriminate a tumor or non the tumor. But this is essentially then radiomics and not liquid biopsy. So I continue on this. This is an interesting story. Maybe we can elaborate in the discussion. I think liquid biopsy phenotyping of cancer can also help us to get an insight in the fundamental biology of the disease. What we did here is based on the microRNAs that we identified as tumor-associated microRNAs. We looked into the public data set, not from the same patients. We didn't sequence the tumor material on this. Whether these microRNAs associate to certain tumors in the tumor tissue, and did the analysis on clinical factors. And indeed, we propose a new microRNA target, by the consensus, we know where the microRNAs bind and which genes are regulated. A microRNA-based target transcriptional subtyping of pancreatic cancer that in some extent aligns with the known transcriptional subtyping of pancreatic, but in some not. So this is a follow-up. We want to see whether that can help us to tailor down a subtype of classical pancreatic cancer that may be drawn to certain genetic interventions because we think it's driven by certain microRNAs, or at least influenced by certain microRNAs than the general classical subtype of pancreatic cancer. Yeah, I wanted to use this opportunity to also showcase technology developments that we do in our department. It has nothing to do at this moment with pancreatic cancer, but I think given the topic, it's of relevance. It's a national funded project with the cooperation partner Candinsa. He's an engineer. It's in the context similarly what we discussed here, pancreatic cancer post-op management in the field of glioblastoma, brain cancer. Here's the same situation that monitoring the disease or the therapy success is expensive. You need machinery like MRI, and it's also insensitive. So a terminology of so-called pseudo-progression emerges where the neurosurgeon and the neuroradiologists are unsure whether the tumor actually responds to the therapy or whether the therapy, in this case, it's a combination of chemo and radiation, induces so much inflammation that this is shown up at the MRI as a similar picture. And, a distinct molecular marker in the blood that associates to tumor could help here. And how this is done in this case in an elegant way, we also focus on microRNAs, is based on a CRISPR-Cas chip. The CRISPR-Cas chip is utilizing not the Cas9 that is prominently known to cut double-strand DNA. We use Cas13a that recognizes single-strand RNAs, such as microRNAs. And the bottom line is more or less that I can go to a next slide. When in a given blood sample, a target is available and is recognized by the custom-made chip with the complementary sequence to this specific microRNA, the double-strand condition is developed and the Cas9 is recruited and activated. Cas13a is activated. And here comes the very big difference. The Cas13 is essentially not selective in the target that is then cutting. It cuts more or less every single-strand RNA that is available in the microenvironment. And here, so to say, the signal quantification is done by the cutting of so-called collateral RNA. These collateral RNAs are immobilized on a chip. And if the Cas13 is activated, here it comes now in the imaging, cuts off the collateral RNA that has a recognition site. In this case, it's a chemical structure that is then under currency uses the glucose to develop reactive oxygen and ion that is then later on transferred into an electric signal. But there can also be a fluorescent signal. So basically, essentially, by cutting off the immobilized collateral RNA and comparing the loss of the signal, you can quantify the amount of activation of the Cas13. And this directly correlates with the known enzyme kinetics with the abundancy of the target. So why this complicated method? Because it's a chip. You don't necessarily even need a PCR. This is a disposable item, essentially. In the mass production, we're talking about 4 to 5 euros. And this can be applied relatively easily, just a blood drop on it. So we are working on this in the context of glioblastoma, and we are currently applying for a project where we want to use this also in the context of pancreatic cancer and doing a side-by-side comparison with the clinical glycan marker CA19-9. I want to also showcase for the people in the interest that our lab has over the last year developed a method to isolate circulating tumor cells. This is only a very brief overview and almost immature image because it's a master student of mine, but I see high level. Then let's move on because the video, I showed a video essentially how this CRISPR-Cas chip worked. We developed the video, but I think we should not go there again. This causes the problem. This is this project we are working on now where we isolate circulating tumor cells from our pancreatic cancer patients. And we used the Illumina DNA metallome array to compare the culture that we established versus the primary tumor. I'm not showing this data here, but what is an important point to mention, and we are going to try to patent this scenario, is that our lab is able now to culture these circulating tumor cells and expanding them and still keeping the epigenetic signature of the tumor piece where we compare the culture with. At this moment, we believe this is a technology that allows us to do drug screening on patients that cannot undergo resection. And we know that when you take a biopsy, that those patients also face problems, including the operation itself. So this is a technology that may help patients that are in an inoperable stage to tailor down their neoadjuvant treatment regime by testing it on the same tumor cells. But this is at a very early stage, and the most important point is that we can grow these isolated cells. This is not in a high fraction, maybe 1 out of 8 patients we are able to do this, but at least from those, and we want to further maturize this technology. And everybody who is interested in this, I think it's a very interesting approach, especially for patients in an operable stage. Imagine if you can tailor down the treatment only to a monotherapy. We know what's the standard of care chemotherapy for pancreatic cancer. It's side effect, and people stop these cycles because of the GI toxicity. If you have one therapy that is efficient and we can identify this in a with such a functional system, it would be very, very good for the patients. I think the time is relatively low, also advanced, but briefly, also from my activities in the field of brain cancer research, it's relatively rapidly emerging in the clinical diagnostics, intraoperative imaging, in particular for differentiating a subtype of the tumor in the OR. People nowadays use Raman spectroscopies. Essentially light, the light touches in the tissue, and the light that is reflected and shining away is altered. And this is so-called the Raman effect, simply described. And this Raman technology is using now in neuro-oncology to discriminate molecular subtypes of the tumor, and maybe in the future help intraoperatively how far one can operate, or even maybe apply intraoperatively targeted drugs. We also worked on this in the field of cell biology. We can discriminate the differentiation status of the cells, so cancer stem cell versus the differentiated progeny, and also pediatric cases versus adult cases, because sometimes it's unclear whether the tumor, even though it's growing in a patient that is young in the neuro-oncology field, is actually a pediatric tumor, or it's more from the biology side considered an adult, and this has relevance for the treatment also option. Yeah, I want to just mention to people use this also to characterize the blood of cancer patients. Here's an example from colon cancer. One has to look very carefully, but there are some peaks in these spectra that associate cancer and discriminate from non-cancer age-matched controls. And yeah, I think this is also an interesting approach, because these Raman technologies nowadays, companies provide this as a ready-to-use tool. It's not a research item anymore. And maybe one can think about this also in the field of liquid biopsy. Yeah, and just as a very brief go over is we use, this is actually the main focus of my lab, cell biology and disease models, not necessarily liquid biopsy. We use also patient-derived model systems to, do target validation, but also in the context of liquid biopsy to validate functional, if those microRNAs are actually coming from a tumor or not, what we discover in our patients' blood samples. And yeah, people who have interest can reach out to Dr. Wartmann. He's my biobank manager. We develop organoids from pancreatic cancer also. And we have a very, very good student that is able to culture these organoids from the pancreatic cancer. We have approximately a growth success rate of 20%, not more. But we have, every week one coming in. So these are organoids that are cryo-stored and can be cultured, and we're using the KRAS mutation detection to discriminate whether it's a tumor or it's not a tumor. Yeah, in one project we focus on stroma co-culture from these organoids to see how these microRNAs shadome is altered. And in a very fascinating project that is funded recently, we mimic the neuro microenvironment. And here we do a full human system. We're going with an IPS that we differentiate to sensory neurons. On the left side, you see the hallmark features of the cells in our lab. They are releasing the substance P. That's essentially like a functional test of functional neurons after you stimulate them with the relevant molecules. Yeah, this is a plan. The project has started 9 months ago to co-culture sensory neurons with primary pancreatic cancer models. Yeah, in summary, I wanted to just say that I think RNA-based blood tests for pancreatic cancer is feasible. It has advantages, as I showed you, coming from the concept of how microRNAs work. It may have even some advantages compared to the very established circulating tumor DNA. But yeah, large-scale trials needed, and probably it must work in combination with other circulating markers, and not as a standalone markers. Topic we don't discuss, reimbursement strategy. I think it's very difficult to bring in the diagnostics and novel diagnostics from a complete new class, essentially. MicroRNA diagnostics reimbursement is almost a life goal, I would say. And as I said, it can also help the basic science when you focus on these liquid biopsy microRNA targets to understand maybe tumor biology better. Yeah, this is a summary of our department. We construct multidisciplinary activities here, cell culture, and various activities in the drug development space. The data-driven research, I think the liquid biopsy research are mostly based on that, what I showed you today. And yeah, we also have some activities in medical device development. If people have interest, can reach out to me. I thank all the funders for these projects, and Professor Kroner again for the trust to bridge such translational projects, and of course collaborators and the patients. I want to also use this opportunity to welcome you to a national congress that we're hosting. Pancreatic cancer will have a dedicated session there. It's the national meeting for surgical research. So all surgical disciplines, including neurosurgery, will gather here. And Professor Kroner is the Congress president, and I'm the Congress secretary. And moreover, people who are interested in, we are recruiting actively talented students or even more senior people. Currently, I have an opening of a postdoc position for 2 years and a PhD student. What you await is an international team, interdisciplinary team. And Magdeburg is an affordable city with only a short duration to Berlin and high quality of life, I believe, for the money you spend here. If you are interested in, please reach out. This is a very, very active recruitment call at this moment. And with this, I thank Transbank for the opportunity to present here.